Good afternoon, class. Today we're going to look at Unit 13, Chapter 21, Part A, the immune system, innate and adaptive body defenses.
A healthy and functioning immune system is the best ally that you and your patients can have, as the immune system's response to infection or injury is the body's own way of healing. For example, if your patient has a cut, the blood clots, and inflammatory cells and plasma proteins enter the area. This is called the inflammatory response and is triggered whenever the body's tissues are injured by physical trauma. The ultimate goal of an inflammatory response is to clear the injured area of pathogens, dead tissue cells, and any other debris so that tissue can be repaired. Once this is accomplished, healing usually occurs quickly. It's essential that you understand signs of a healthy immune response so that you can identify any cases where the body does not heal on its own. In cases of a severe infection, pus, a mixture of dead tissue cells and pathogens, may accumulate in the wound. If the inflammatory response fails to clear the area of debris, the body may wall off a sac of pus and form an abscess. At this point, the patient will need medical care to assist the immune system's response, and the abscess may need to be surgically drained before healing can occur. Having a solid understanding of the body's response to infection and injury will enable you to properly care for your patients.
The immune system provides resistance to disease. It's made up of two intrinsic systems, the innate or non-specific defense system and the adaptive or specific defense system. The innate constitutes the first and second lines of defense, whereas the adaptive is the third line of defense. So the first line of defense has to do with the external body membranes, so the skin and mucous membranes. Whereas the second line of defense involves antimicrobial proteins, phagocytes, and other cells that inhibit the spread of invaders. Inflammation is actually the most important mechanism in the second line of defense. In the adaptive defense system, the third line of defense attacks particular foreign substances, and it takes longer to react than the innate defense system. The immune system is a functional system rather than an organ system. The innate and adaptive defenses are intertwined. Both release and recognize many of the same defensive molecules. Innate defenses do have specific pathways for certain substances, and innate responses release proteins that alert cells of the adaptive system to foreign molecules. Figure 21.1 is a simplified overview of innate and adaptive defenses. So the innate defenses, we have the surface barriers, that's the first line of defense, that's the skin and mucous membranes, and the internal defenses, that's the second line of defense. There's phagocytic cells, natural killer cells, the inflammatory response, antimicrobial proteins, and fever. In the adaptive defenses, there's the B cells and T cells. B cells are humoral immunity, whereas cellular immunity is the component of cellular immunity is the T cells. Part 1, innate defenses. Okay, so innate system uses the first and or second lines of defense to stop attacks by pathogens. Pathogens are disease-causing microorganisms. So it could be a virus, it could be a bacteria, it could be protists, it can be fungus. So you'll see more specifically in microbiology what the organisms are. Section 21.1, the first line of defense, surface barriers. So surface barriers are our skin and mucous membranes along with their secretions, so the skin is a physical barrier to most microorganisms as long as it remains intact. Keratin on the surface of the skin is resistant to weak acids and bases, bacterial enzymes, and toxins. And the mucous membranes provide similar mechanical barriers. The skin and mucous membranes produce protective chemicals that inhibit or destroy microorganisms. They produce acid, so the skin there's this acidity of the skin and some mucus secretions that inhibit growth, and it's called the acid mantle. There are some bacteria that are able to thrive in these conditions, but they do not make us sick. And also there's some enzymes that are produced, like lysozyme, that is found in saliva. It's a component of saliva, and lysozyme, is important because it inhibits gram-positive bacteria. Respiratory mucus, lacrimal fluid kills many microorganisms. Also, the enzymes in the stomach kill many microorganisms, as well as the acidity in the stomach, in fact. Mucin is a sticky mucus that lines the digestive and respiratory tract, and this has the function of trapping microorganisms. Defensins, these are antimicrobial peptides. Peptides are small proteins that inhibit microbial growth. And other chemicals like lipids in sebum and dermicidin in sweat are toxic to some bacteria, at least to those bacteria that could potentially make us sick. The system also has modifications to stop pathogens. Mucus-coated hairs in the nose will trap inhaled particles. Also, the cilia of the upper respiratory tract will sweep dust and bacteria-laden mucus toward the mouth. We already talked about the ciliary escalator. So cilia that is present in the trachea. We've looked at the cross-section of the trachea. Surface barriers breached by nicks or cuts trigger the internal second line of defense that protects deeper tissues. Table 21.1 summarizes the first line of defense. That is basically surface membrane barriers. So you can see the category or associated elements and the protective mechanism. Section 21.2, second line of defense, cells and chemicals. So the innate system is necessary if microorganisms invade deeper tissues, and this includes phagocytes, natural killer cells, the inflammatory response, which includes macrophages, mast cells, white blood cells, and inflammatory chemicals, and antimicrobial proteins, such as interferons and complement proteins, and of course, fever. Many second-line cells have pattern recognition receptors that recognize and bind tightly to structures on microbes, disarming them before they do harm. One class is known as the toll-like receptors that play a central role in triggering the immune response. Humans have 11 different toll-like receptors, and each recognizes a particular class of attaching microbe. Let's start looking at phagocytes. So phagocytes are white blood cells that ingest and digest foreign invaders. Neutrophils are the most abundant phagocytes, but die fighting. They become phagocytic on exposure to the infectious material. Another type of phagocyte is called are the macrophages. These develop from monocytes and are the chief phagocytic cells. The most robust phagocytic cell, in fact. There are free macrophages. These wander through tissue spaces. Example, the alveolar macrophages that we've already discussed when we looked at the lungs. And fixed macrophages, these are permanent residents of some organs. For example, stellate macrophages like the liver and microglia of the brain. Figure 21.2a, phagocytosis. So here we're looking at a macrophage. That's that big purple cell, and it's about to engulf a rod-shaped bacteria, and that's in green. So they send these extensions that surround the bacterial cell, and then will engulf it, and it's going to ingest it and break it down. Let's look at the process of phagocytosis. This is the process that starts when the phagocyte first has to recognize and adhere to the pathogen's carbohydrate signature. So it first recognizes that it's foreign, and then it's going to adhere to it. So some microorganisms have external capsules that hide their surface carbohydrates, helping them evade phagocytosis. Opsonization is what the immune system uses. So it's when the immune system uses antibodies or complement proteins as opsonins that will coat the pathogen, and it acts as handles for phagocytes to grab onto, really enhancing phagocytosis. Figure 21.2b. So the phagocyte adheres to the pathogens or debris using receptors. Second, these cytoplasmic extensions or pseudopods bind to and engulf the particle in a vesicle called a phagosome. And thirdly, the phagosome fuses with a lysosome, forming a phagolysosome. And then the phagolysosome is acidified, and lysosomal enzymes digest particles. Remember from a previous chapter, we talked about how lysosomes took care of intracellular digestion. And anything that's indigestible and residual waste is exocytosed from the phagocyte, is discarded to the outside. So here we can see the phagocyte forms the pseudopods that eventually engulf the particles, forming a phagosome. And then eventually the phagosome is going to fuse with a lysosome, which is going to hydrolyze the bacterium in this case, chop it up into pieces, and then anything that's discarded is going to be exocytosed. I was a little bit ahead of myself in the previous slide, but here you can see in step 3, the lysosome fuses with the phagocytic vesicle, forming a phagolysosome. And in 4, toxic compounds and lysosomal enzymes destroy the pathogens. And then sometimes exocytosis of the vesicle removes indigestible and residual material. Some pathogens are not killed with acidified lysosomal enzymes. An example is the tuberculosis bacteria. Helper T cells trigger the macrophage to produce respiratory burst, which kills the pathogens resistant to lysosomal enzymes by releasing cell-killing free radicals, producing oxidizing chemicals like hydrogen peroxide, and increasing the pH and osmolarity of the phagolysosome. Defensins in neutrophils also help by piercing the membrane of the pathogen. And then, of course, it's going to spill out its contents. A third component of the second line of defense are the natural killer cells. These are non-phagocytic. They're large granular lymphocytes that police blood and lymph. They can kill cancer and virus-infected cells before the adaptive immune system is activated. So that's their specialty, cancer and virus-infected cells. They attack cells that lack the cell surface receptors, and they kill by inducing apoptosis or programmed cell death in cancer cells and virus-infected cells. They secrete potent chemicals that enhance the inflammatory response. Look at inflammation. So tissue response to injury. So inflammation is triggered whenever body tissues are injured. Injuries can be due to trauma, heat, irritating chemicals, or infections by microorganisms. The benefits of inflammation are, first, very important, it prevents the spread of the damaging agents. It disposes of cell debris and pathogens. It also alerts the adaptive immune system and sets the stage for repair. The 4 cardinal signs of acute inflammation are redness, heat, swelling, and pain. Sometimes a 5th sign, impairment of function, is seen if movement or use of the area is hampered. Stages of inflammation. So there's the inflammatory chemical release, vasodilation and increased vascular permeability, and phagocyte mobilization. Inflammatory chemical release. So chemicals are released into the extracellular fluid by injured tissues or immune cells. An example are histamine, that's released by mast cells, and it is a key inflammatory chemical. Other inflammatory mediators besides histamine are kinins, prostaglandins, cytokines, and if the pathogens are involved, complement. And these all cause vasodilation of local arterioles, all make capillaries leaky, and many attract phagocytes to the area. Table 21.2 is a summary of the inflammatory chemicals, such as histamine, kinins, prostaglandins, complement, and cytokines, their source and physiological effects. So let's look at vasodilation and increased vascular permeability. Vasodilation causes hyperemia, which is congestion with blood, which leads to redness and heat. Increased capillary permeability causes exudate, where a fluid containing clotting factors and antibodies to leak into the tissue, and this results in the local swelling or edema. Swelling also pushes on nerve endings, and this will result in pain. Pain can also result from release of toxins from bacteria or released prostaglandins and kinins. Benefits of edema are surge of fluid in the tissue sweeps the foreign material into the lymphatic vessels for processing in lymph nodes, delivers clotting proteins and complement to the area. Clotting factors form fibrin mesh that acts as scaffold for repair, and the mesh also isolates the injured area so that the invaders do not spread. Phagocyte mobilization. Neutrophils flood the area first, macrophages follow. If inflammation is due to pathogens, complement is activated and the adaptive immunity elements will arrive. So steps for phagocyte mobilization. Leukocytosis is a release of neutrophils from bone marrow in response to leukocytosis-inducing factors from the injured cells. Margination is when the endothelial cells of the capillaries in the inflamed area project cell adhesion molecules into the vessel lumen that grab onto passing neutrophils, causing them to slow and roll along, clinging to the vessel wall. Diapedesis is when neutrophils flatten and squeeze between the endothelial cells, moving into the interstitial spaces. The fourth step for phagocyte mobilization is chemotaxis, where the inflammatory chemicals act as chemotactic agents that promote positive chemotaxis of neutrophils toward the injured area. As attack continues, monocytes arrive later. 12 hours after leaving the bloodstream, they are transformed into macrophages. These late arrivers replace the dying neutrophils and remain for cleanup prior to repair. One point 3, phagocyte mobilization. So in step 1, leukocytosis, the neutrophils enter the blood from the bone marrow. 2 is margination. Notice how the neutrophils cling to the capillary wall. 3 is diapedesis. The neutrophils will flatten and squeeze out of the capillaries. And in step 4, chemotaxis, neutrophils will follow the chemical trail. So it's a positive chemotaxis because they are orienting themselves towards the those inflammatory chemicals and the bacteria. Figure 21.4, events of acute inflammation. Okay, so here we have a cut. Okay, tissue repair. So release of inflammatory chemicals like histamine, complement, kinins, and prostaglandins will cause arterioles to dilate, increase capillary permeability, and these will attract neutrophils, monocytes, and lymphocytes to the area through positive chemotaxis. And these will migrate to the injured area, and we will see what we talked. So this is like leukocytosis, margination, diapedesis, and phagocytosis. And then because the arterioles dilate, there's local hyperemia, which increases the blood flow to the area that will cause the heat and the redness. And locally increased temperature increases the metabolic rate of the cells. Also pain and also swelling because of the leaked protein-rich fluid in the tissue spaces. So this is just a summary of what we've seen so far. Clinical homeostatic imbalance 21.1. Pus is a creamy yellow mixture of dead neutrophils, tissue cells, and living or dead pathogens. An abscess is when collagen fibers are laid down, walling off the sac of pus, and may need to be surgically drained. Some bacteria, such as the tuberculosis bacterium, resist digestion by macrophages and remain alive inside. They can form tumor-like growths called granulomas, the area of infected macrophages surrounded by uninfected macrophages and outer capsule. Bacteria may remain inactive forever, or if the person's immunity decreases, they may break free, become activated, and then cause disease. Okay, so let's talk about antimicrobial proteins. So antimicrobial proteins enhance the innate defense by attacking microorganisms directly or hindering the microorganism's ability to reproduce. The most important antimicrobial proteins are interferons and complement proteins. Interferons is a family of immune modulating proteins. Cells infected with viruses can secrete interferons that warn healthy neighboring cells. So these are very important also in the fight against COVID, in fact. Interferons enter neighboring cells, stimulating production of proteins that block viral reproduction and degrade viral RNA. Interferon type A, alpha, and type B, beta, work this way. Interferon A and B also activate the natural killer cells. Interferon gamma, also called immune interferons, this is secreted by the lymphocytes, has widespread immune mobilizing effects. It activates the macrophages. Interferons activate natural killer cells in macrophages, so they indirectly fight cancer. Artificial interferons are used to treat disorders such as hepatitis C, genital warts, and multiple sclerosis. Figure 21.5, interferon mechanism against viruses. Okay, so here we have a virus that comes in with its viral nucleic acid. The virus enters the cell and replicates. Interferon genes are switched on, and the cell produces interferon molecules. And then what happens is that interferon binding stimulates the cell to turn on genes for antiviral proteins, and the antiviral proteins block viral reproduction. So this is in fact what happens with COVID-19. So when it comes in, the virus is bringing its RNA, nucleic acid, invades the cell, and in people that have a good interferon response, they manage to block viral reproduction. Complement is another example of antimicrobial proteins. The complement system consists of about 20 blood proteins that circulate in blood in inactive form. It includes proteins C1 to C9 and factors B, D, and P, and regulatory proteins. It provides the major mechanism for destroying foreign substances. Activation enhances inflammation and also directly destroys bacteria. It enhances both innate and adaptive defenses. The system can be activated by 3 different pathways. The first one is called the classical pathway, where antibodies first bind to the invading organisms and then bind to complement components, activating them. Double binding called complement fixation. Once the initial complement proteins are activated, an activation cascade is triggered. The lectin pathway. Lectins are produced by the innate system to recognize foreign invaders. When lectin is bound to specific sugars on the foreign invaders, it can also bind and activate complement. And the third pathway is called the alternative pathway. This is when a complement cascade is activated spontaneously when certain complement factors bind directly to a foreign invader. Lack of inhibitors on the microorganism's surface allows the process to proceed. Each pathway involves activation of proteins in an orderly sequence. Each step catalyzes the next in a cascade fashion. Each pathway converges on C3, which cleaves into C3A and C3B. Splitting C3 initiates the common terminal pathway that enhances inflammation, promotes phagocytosis, and causes cell lysis. Cell lysis begins when C3b binds to the target cell, triggering insertion of complement proteins called membrane attack complex into the cell's membrane. The membrane attack complex forms and stabilizes a hole in the membrane of the microbe, causing an influx of water and lysis of the microbe. C3b also causes opsonization. C3a and other complement protein cleavage products amplify inflammation by stimulating mast cells and basophils to release histamine, attracting neutrophils and other inflammatory cells. 0.6 complement activation. So here are the three different pathways: the classical pathway, the lectin pathway, and the alternative pathway. So the classical pathway, so all three pathways converge at C3 to initiate the final activation of complement together with other complement proteins and factors. C3 is split into two active pieces, C3a and C3b. Opsonization, remember, is that process when the pathogen is coated, and this enhances phagocytosis. So that's due to the action of C3b. C3a instead will stimulate inflammation. So it stimulates histamine release, increased blood vessel permeability, attracts phagocytes by chemotaxis, and etc. All those steps in inflammation. And then all together there, when you have MAC, membrane attack complexes form when C3b activates other complement components, and this occurs only on the surfaces of certain target cells, usually bacteria. MACs insert into the target cell membrane, creating these pores. Okay, so they're pretty much punch holes into the membrane. And that will cause lysis of the target cell by allowing water to enter and the cell contents to leave. Fever is an abnormally high body temperature that is systemic. Is a systemic response to invading microorganisms. Leukocytes and macrophages exposed to foreign substances secrete pyrogens. Pyrogens act on the body's thermostat that is located in the hypothalamus of the brain, raising the body temperature. Benefits of moderate fever: it causes the liver and spleen to sequester iron and zinc needed by microorganisms. So basically the liver is going to hide these two minerals so that they're not available to microorganisms. And also moderate fever is going to increase the metabolic rate, which increases the rate of repair. Table 21.3, this summarizes the second line of defense: innate cellular and chemical defenses. We can see phagocytes, natural killer cells, inflammatory response, antimicrobial proteins, and fever and their protective mechanism.